Dear students, after completing the fertilization types of fertilization that we have completed external fertilization and internal fertilization, we are just going to start with the event that occurs after the fertilization, and all those events are called as post fertilization events. The post-fertilization events are the events just after occurs completes after the fertilization, found after the fertilization. Here we have to discuss two different events. One is zygote and second one is embryogenesis. So one by one we are just going to start with. The first one we are just taking the zygote. As we all know, the fertilization is fusion of gamete. Gametes are usually haploid in nature, which is represented as N. By the fusion, the gametes N and N will combine to form a diploid structure, that is 2N, and this diploid structure is called as zygote. So this is the fate of all the organisms कि after fertilization will lead to the formation of diploid zygote, a diploid structure. Yesterday only we have just concluded that objective of the fertilization to restore the diploid whatever is being lost during the gametogenesis. So here zygote is a universal event or universal structure in all the sexually reproducing organism. One more important point I wish to put in your notice that zygote is a kind of connection, or zygote is a vital link. Vital means a living structure or living link that ensures the continuity of species between the organism, between generations. So are you getting the meaning of this point? So first of all we must understand that whenever the fertilization process occur, the fertilization will always result in the fusion of haploid gametes, and due to the fusion of that haploid gamete, what will happen? A diploid structure form. That diploid structure is called as zygote, and this is why whenever the fertilization or whenever the syngamy will be there, the organism will have to form the zygote. This is what we are saying, that formation of zygote is universal event in all the organisms if they are just performing the sexual reproduction. Secondly, the zygote is a kind of living link. Living link means to say that zygote is a living structure which is being formed after the fertilization. So it is a living link which ensures the continuity of the germplasm, which ensures the continuity of the genes, which ensures the continuity of the character from one generation to the next generation. This is carrying the character from one generation to the next generation. This is what two very important question. Zygote is a vital link and zygote is a universal structure which is going to ensure the continuity of the properties or continuity of characteristic between two generation, from mother to the daughter individual, from father to the daughter individual. I think it is very much clear to you all. And very next we are just going to understand some of the organisms, those who are having the external fertilization and external development, means when they are performing the external fertilization occurs outside of the body in the aquatic medium, like algae, fungi, They are having external development. They produce a very specific condition. Suppose zygote is there. That zygote is being laid outside, or zygote is developed outside of the body there in the aquatic medium. The aquatic medium is having full of the danger to by which the zygote can get desiccated, can get damaged, can get destroyed. To make the separate, to make safe against such kind of the danger, to make the harness safe against such kind of the danger, these harness develop a kind of protective covering, that is thick resistant covering around the zygote. Inside that zygote is there, nucleus is there. So are you getting my point? Whenever the external fertilization is there, zygote is lying outside. So at that time, to make them protect against the harsh, dangerous condition by which the zygote can get damaged, zygote can get desiccated. So to avoid such kind of the situation, the zygote develop itself a thick resistant wall around itself, which provide them protection against the desiccation and damage. The zygote remain inactive. Inactive means we can say under the resting phase. Until and unless the conditions are getting favorable for the development. When conditions get favorable, it get germinates and will begin the formation of embryo. That means embryogenesis will begin. So are you clear with me? The zygote is a kind of a structure which is being produced after the fusion of gamete. Zygote is the diploid structure. So whenever the sexual reproduction is there, all the sexual reproduction always conclude with the formation of zygote. This is what we are saying, ki all the organisms which are performing the sexual reproduction will have to produce a universal structure, that is a zygote. And secondly, zygote is only structure, zygote is the structure which is present in all the sexual reproduction, sexually reproducing organisms, by which they are transferring the characteristic from one generation to the next generation. Secondly, we have discussed so many things about the zygote. One more important question is there, ki somewhere the zygote is being laid outside, development of zygote occurs outside of the body there, in the cases of external fertilization, that what we have understood yesterday only, in the previous lecture only. So if such kind of the situation is there, so zygote may have a danger to get damaged from the external agencies, may get desiccated, may get destroyed. To avoid such kind of the situation, zygote develop itself a thick resistant covering around it and remain inactive inside until and unless it gets the favorable condition. As soon as it gets the favorable condition, it is going to germinate and will start the development of embryo. This is what called as embryogenesis. One more point we have to take care: the development of zygote is different in different species, different organism, depending upon the life cycle. So let's move on to the next half of the topic, next half of next event of the post fertilization event, that is embryogenesis. So next event is embryogenesis. What is embryogenesis? Once again, if we are just going to the etymology, embryogenesis, formation. Embryogenesis means formation of embryo, means the zygote will convert in form of embryo, that is called as embryogenesis. Are you getting my point? The zygote which is formed and is a diploid structure is getting converted in form of a diploid embryo called as embryogenesis. How embryogenesis occurs? Embryogenesis is completely a process of division and differentiation. Last year only in class 11th you might have gone through the process of division and differentiation. The division occurs is mitosis, and after mitosis differentiation is there. What is the meaning of division and differentiation? Division, mitosis means always 2n to 2n cell will be produced, means diploid to diploid cell will be produced, there will be no reduction in the number of chromosome. And differentiation means after a continuous repeated mitotic division there will be great number in increase, there will be great increase in the number of the somatic cell or diploid cell. Now these cell will start moving toward their organ specific area, their cells will start getting converted in form of their particular type of the structure. This is called as differentiation. Basically, if you have remembered, differentiation is the process of conversion of temporary cell to permanent cells. In class 11th only you have gone through that. Temporary means the cell which have capacity of division, and permanent they lost the division capacity. Are you getting my point? So two basic event occurs during the embryogenesis or embryogeny: the division and differentiation. The division is basically a kind of mitotic division where a diploid cell is going to produce the diploid cell. Only the change is there, the great number of new cells are increased thereon. After increasing the number of cells, the cells are going to convert in form of the permanent cell as per the construction of the particular structure in the organism body. This is called as differentiation, means the differentiation is conversion of the dividing cell into the non-dividing cell. So how embryogenesis occurs in animals and how it occurs in plants. So first of all we are just observing in animals. Suppose a diploid cell 2n, that is zygote, it will undergo in the series of mitotic division. That process is referred as cleavage. That process we'll discuss there later in the next chapters, where it will form the group of new cells. These group of new cells are called as morula. Now morula stage will develop a centrally liquid filled cavity, central cavity therein, and all the cells are going to be shifted onto the peripheral layer. These cells are called as blastomeres. You knows well, last year only we have just gone through the structures, we have gone through the concept. The blast cells are the cells which are able to generate the new structures. Cavity filled inside, cavity present inside is now known as blastocele, and the structure formed here is called as blastula. In human this is referred as blastocyst. Later on The process of differentiation began, and this embryo develops three different layers inside. The outer one, the inner one, and the middle one. The outer one is called as ectoderm. The inner one is called as endoderm. The middle one is called as mesoderm. These ecto, meso and endoderms are the germ layers. These germ layers have responsibility to produce the new structures, and the stage is now called as gastrula. Formation of gastrula is called as gastrulation. After formation of gastrula, after formation of germ layer, now organogenesis begin. Organogenesis is the formation of organs. Formation of organs are specific role taken over by the ectoderm, mesoderm and endoderm. These structures or these cells or cells of these regions will move toward their organ specific areas, will convert in form of a particular organ. Like that, if ectoderm has to give rise the nervous system, so nervous system will be produced by the ectoderm. If mesoderm is going to produce the visceral organ, so this will produce the visceral structure.